Okay, good afternoon everyone. Welcome to lecture 50 of BMS 300, right? And we're going to start with part 1 of the urinary system. And as usual, when we start a new organ system, I'd like to begin with an integration slide so that you know how the functioning of the urinary system is dependent on other systems and how it can influence other systems as well, right? So we see that the urinary system has many functions, but one of the major functions is to filter the blood. And when you think about the blood composition, we see that it's mainly made up of water, and there are also some proteins found in the blood, and we did discuss how the synthesis of those proteins will happen during central dogma. And in blood being filtered, we notice that there are a number of transmembrane proteins which will play a role in the exchange of substances, whether they are being absorbed back into the bloodstream after it's being filtered, or if there are secretions where if something did not get filtered into the filter out of the blood, it can be secreted back in. So we'll see that there are a number of transmembrane proteins we'll be considering to see how the urinary system functions. And then when we talk about the various cell junctions, we will specifically talk about tight junctions, how they are found holding the epithelial cells in the nephrons where the urine will be formed. But we see that among the cells which are being held together, it's mainly held by tight junctions, and we'll see that some of these tight junctions have pores in them where selectively certain substances can get in between the cells. As far as the various tissues are concerned, we'll talk about different types of epithelial cells depending on which part of the kidney or the parts of the urinary system you're considering, because different types of cells will mean that there is different types of functions being carried out. We also look at the role of the smooth muscles which are found in the urinary system, and we'll also see where the connective tissue is found as well. As far as the integumentary system is concerned, we find that the skeletal system, endocrine system, and the digestive system function can be all linked together when we consider the synthesis of vitamin D. I told you that vitamin D in its active form is produced in the kidneys, right? But we find that vitamin D will have an impact in the digestive system to help in the absorption of calcium, which is finally going to be incorporated into the bones. And because all of these things are held together in the functioning of vitamin D, I would like to link these organ systems using that. And then moving on to the nervous system, if there is a drop in blood pressure, the kidney will detect that, and then signals can be sent to the central nervous system, and accordingly there will be responses made, and we'll consider this later in the week. And we find that there are different types of smooth muscles which line with the various smooth muscles which line the urinary system as well, and we talk about their functions. And the cardiovascular system is what will give pressure to the blood so that it can be filtered. And we find that the respiratory system is not the only one which maintains the blood pH. We also find that the urinary system can play a role in the exchange of various components, and that can help in maintaining the blood pH as well. And finally, we find that if there are some white blood cells showing up in the urine, it may be indication of urinary tract infection. And so we find that components of the immune system being found in the blood can also be used as a diagnostic tool. So this is how all of the organ systems are connected together as far as the urinary system is concerned. But now let's move on to have a quick overview of what we'll be discussing today. So we'll start by talking about the various parts of the urinary system, and I will highlight the difference between the male and the female urinary system, specifically to point out why females have a higher risk of urinary tract infection. And then we'll talk about how the blood is going to flow through the kidneys. We'll see how the blood vessels will branch repeatedly, but we also will notice how the blood will be carried through the nephrons. So the nephrons are the basic structural and functional unit of the kidney. Whatever happens at the nephron level will translate to the functioning of the kidney as a whole. And having discussed about the parts of the nephron, you'll see what are the 3 steps involved in urine formation. So urine formation is a pretty complex process, but we can consider it in 3 simple steps, and then we'll get into the details. So the first step is going to be the filtration of the blood. And notice that the blood has a lot of plasma that will be filtered into these tube-like structures, and then there will be a whole lot of reabsorption of various substances from the filtrate back into the bloodstream, because it's a very small percentage of this filtrate which eventually turns into urine. And then the third step is secretion. If something did not get filtered out initially into the substance, into the fluid which turns into urine, those unfiltered substances can be secreted back into the filtrate, which will be excreted as urine. So these are the 3 steps in urine formation. So with that, let's consider what are the major organs of the urinary system. So we see that the kidneys are the major organs of the urinary system, and they will play a role in filtering the blood and in producing the urine. So this urine which is produced in the kidneys will be sent through the ureters. So these are tube-like structures, and each kidney will have a ureter, and that will carry the urine to the reservoir, which is the urinary bladder, where the urine will be stored. And then once the signal is sent from the central nervous system, the urine will be sent out through the urethra to the external environment. And so we find that in males and in females, the urethra will be used to transport out urine, but in males, semen can also be transported out through the urethra. So I find that it's very easy for folks to confuse between the names ureters and urethra, and to forget what their functions are. So one helpful hint I have for you is that think about urethra as being the tube-like structure which throws the urine out of the urinary system to the external environment. So urethra will help to throw out the urine to the external environment. Okay, so I hope that helps you. So now let's move on to talk about each of these organs in the urinary system. So starting with the kidney, we see that these are bean-shaped organs which are paired. So you have a left kidney and a right kidney. And what this indicates is that there seems to be a redundancy in the function, right? And the relevance of this is that if the functionality of the 2 kidneys were to drop down to 25%, death will occur very soon. And the problem is, as there is failure in one kidney, there is no symptoms at all, right? And it makes sense why the body needs to have a redundancy in function, so that if even one kidney fails, the other one can keep functioning. And there are people who can survive with just a single kidney as well. So reasons for complications in the kidney can arise from high blood pressure, and we see that there are a number of blood vessels in the kidneys, and these blood vessels can be very thin, and so if there is a abnormally high blood pressure, it can have an impact on those blood vessels, and it can actually affect their integrity. As far as diabetes is concerned, you're all aware of how advanced glycation end products can lead to the clogging up of the blood vessels in the kidney due to plaque formation. Okay, so that's something you should be familiar with. We also find that the right kidney is going to be slightly lower than the left kidney because it will be crowded out by the liver. You remember that the liver also does some crowding out of the lung as well. Is it the left lung or the right lung? Right lung, that's right, because the left lung is going to be crowded in by the heart, right? It has the cardiac notch, right? So now let's take a closer look at how the kidneys are. Oh wait, before that, so if the kidneys are taken out of the body, right, how do you identify which is the left and the right kidney? What is the unique feature? the left and the right kidney once it's outside the body? Nobody knows? Okay, I don't know either, right? There's no way to to distinguish one from the other. Anyway, what I need to do to keep people engaged, right? So anyway, so we see that this kidney's got an outer capsule, as you see right here. This fibrous capsule is going to surround a layer of perirenal fat. Okay, here's the perirenal fat shown here, and the purpose of this kind of arrangement is to help absorb any sort of shock which the kidney may encounter. And so in the abdominal cavity, we find that there is this membrane known as a peritoneum, which is going to surround the various structures in the abdominal cavity. And if any of you have the opportunity to have done dissections, or you do dissections in the future, and you, let's say, open the stomach of a rat, the abdominal region, and you take out the small intestines, the stomach, and then the liver, you will notice that in the existing cavity, it looks like the kidneys are actually stuck behind wallpaper, right? Because they're going to be found behind the peritoneum, and so they're just held together to the back of the abdominal wall. And so this, because they're found behind the peritoneum, they're called retroperitoneal. So now let's take a closer look at the internal anatomy of the kidney. So when you think about the cross section of the kidney, you can divide it into two parts: the outer part, which is the renal cortex, right, and then the inner part, which is the renal medulla, right. And so we will see that the components which are found in the cortex will differ from the components found in the medulla. That's why they look so different, even from an anatomical perspective. So in the medulla, you'll notice that there are a number of these pyramid-like structures called renal pyramids, as the name indicates. And each of these pyramids, they're going to be separated by narrow columns, which are known as renal columns. So the names really match up with what you're seeing. Okay, and each of these renal pyramids, they will have like a pointed apex, right? And urine is going to be formed within the renal pyramids, and it will flow to the renal papillae, and from the renal papillae it's going to move into these tube-like structures called the minor calyx. Multiple minor calyx will fuse to form a major calyx, and multiple major calyces will fuse to form this renal pelvis, which is this funnel-shaped structure. And the renal pelvis will eventually lead to the ureters, which will carry the urine from the kidneys. Okay, so we see that the urine will flow all the way from the papillae, which is the apex of those pyramids, down to the ureter. So now let's move on to consider the structure of the ureters. So we see that the ureters are also paired, because each kidney has one ureter arising from it, and its purpose is to convey the urine all the way to the bladder. And we see that these are called as muscular tubes because they have a layer of smooth muscle inside them, as I will show you in a second. But notice that even these ureters are going to be retroperitoneal. They are found behind the peritoneum, and they will enter the bladder at the base of the bladder, right? They will terminate at the base of the bladder, and the region where they enter in will lead to this opening known as the ureteric orifice. So orifice is referring to a hole, because these are holes found at the end of the ureters. They are called the ureteric orifices. So a cross-sectional view of the ureters will reveal that the lumen is going to be star-shaped, right? And this star-shaped lumen is going to be lined by transitional epithelial cells. So we will find that a cross-section will reveal three distinct layers. The first layer you see will be the transitional epithelium. I told you the transitional epithelium are these cells which are capable of distending and returning back to their normal shape. So when they are not distended, they are going to be dome-shaped. When they are stretched out, they will turn flat. And we will find this transitional epithelium in various parts of the body which are capable of stretching. So these transitional epithelial cells, they are found under a layer of connective tissue, specifically the areolar connective tissue, and that's called the lamina propria, right? And then superficial to the lamina propria, you have this muscularis layer where you have a bunch of smooth muscles, and this is why it's called as a muscular tube. And more superficial to the muscularis layer, you have adventitia, which is more connective tissue, which will provide attachment for adipose tissue, fat, blood vessels, and nerves as well. So you will find these three layers as you consider other parts of the urinary system as well. So now we move on from the ureters to the bladder, and we see that the bladder is going to be highly distensible, right? So what type of epithelium would you find here? Same thing, transitional epithelium, right? So because it's highly distensible, it can store typically around half a liter of urine typically. But then let's say you're put in a situation it can accommodate up to a whole liter of urine. So the example I like to give is imagine you're on the way to the airport and you're running late, you badly need to use the bathroom, but if you know you stop, you will miss your flight, right? So thanks to your urine, thanks to your bladder which can distend, you can continue driving and make your flight, and then you can relieve yourself, right? So this is one of the benefits of the bladder being highly distensible. So now let's see how the bladder is positioned in the male versus the female urinary system. So in males, it's going to be found superior to the prostate gland. So here's the prostate gland here, here's the bladder. Cross-sectional view shows here's the prostate and here's the bladder as well. And we find that in females, it's going to be positioned slightly differently because it will be found inferior to the uterus, right? And it's also found anterior to the vaginal openings. Here's the vagina and here's the urethra, and so the bladder will be found anterior to the vagina. Now the relevance of the bladder being immediately inferior to the uterus is that during pregnancy, as the fetus continues to develop, and specifically in the third trimester, the fetus will put pressure on the bladder, and that is why pregnant women will have such an urge to urinate so often, because the fetus keeps putting pressure on the bladder. And so a cross-sectional view reveals that this is also got transitional epithelium because the bladder is capable of distending as it fills up with the urine. So now we move on from the bladder to the urethra, the tube-like structures which throws the urine out to the external environment. And notice that in both males and females, it's a muscular tube which will transport urine out of the body, right? And we see that it's going to end in what is known as the external urethral orifice. So orifice is referring to an opening, and this is the external urethral orifice you find at the end of the urethra. So let's talk about some of the differences between the male and the female urethra, and it is some of these reasons which will tie into why females have a higher risk of urinary tract infection. So in females, it's pretty short, it's 3 to 4 centimeters in length. It's going to have the orifice or the opening to be anterior to the vaginal opening, and it will also have a sole purpose of excreting urine, right? Whereas in males, the urethra is going to be much longer, around 20 centimeters in length, and these 20 centimeters can be divided into 3 specific regions, right? And I'll tell you what these 3 regions are. The first region is the prostatic urethra, which is going to pass through the prostate gland, and the second region is the membranous urethra. This is just a very narrow region as the urethra exits the prostate gland and it moves into the shaft of the penis. And in the shaft of the penis, the urethra is known as the spongy urethra. And we find that the external urethral orifice is going to end at the glans penis as shown here. And I mentioned previously that the urethra in males transports both semen and urine as well. And we will actually take a closer look at why both semen and urine will be transported through the urethra when we look at the male reproductive system later in the next couple of weeks. So I keep mentioning the type of epithelial cells found in different parts of the urinary system because the function of that region will be influenced by the type of epithelial cells, right? So to help you think in that regard, I want you to consider this poll everywhere question. Which type of epithelial cell would you find in the external orifice of the urethra? So which type of epithelia would you find in the external orifice of the urethra? Would you find simple cuboidal epithelia, simple squamous epithelia, stratified squamous epithelia, or none of the above? Think about structure function relationship. Okay, most of you picked stratified squamous epithelia, and that is correct, because we know that the stratified squamous epithelia is necessary because that part of the body would have more physical abrasion, so it needs to have a protective function given to it. That's why it has stratified squamous epithelia. So cross section of the urethra will reveal this branching lumen, and notice that the type of epithelia found in the urethra is going to be variable, right? It really depends on which part of the urethra you are looking at. So closer to the bladder, we find that there will be transitional epithelium, because we know the bladder is capable of distending, and that will have transitional epithelium. So the part where the urethra connects to the bladder will have transitional epithelium. However, we see that most of the urethra will be lined by pseudostratified columnar epithelia in both males and females, and we see that this pseudostratified columnar epithelia can have cilia, which can help propel the urine downwards. And as you all mentioned, that the external orifice will have the stratified squamous epithelia because it's more prone to physical abrasion. And notice that this transitional epithelia, or whichever type of epithelia, will be supported by a layer of connective tissue, which is going to form the lamina propria, and then there will be this muscularis layer showing you that there are smooth muscles to propel the urine out of the body, and then there will also be this adventitia layer, which is more connective tissue, which is going to support the surrounding structures like the nerves, blood vessels, and so on. So now let's talk about why females have a higher risk of urinary tract infection. So some of these reasons could be anatomical, others are based on behavior and also physiological reasons as well. So the first reason I mentioned is an anatomical reason. We see that the length of the female urethra is much shorter, so if the bacteria were to invade this region, they have a much shorter distance to travel, right? And that's why this can increase the risk of urinary tract infection in females. Also, the proximity to the rectum means that fecal contamination can also easily reach this urethral orifice, and that's why the recommendation is to wipe from front to back after defecation. We find that during sexual activity, because the urethral opening is anterior or in front of the vagina, we find that germs from the vagina can also easily infiltrate this region, and it can increase the risk of urinary tract infection. Now, there are some contraceptives which can also play a role in increasing this risk, and the reason is contraceptives which are containing spermicides like diaphragms or condoms with spermicides in it, they will not only kill the sperm but also beneficial bacteria, thereby leading to the proliferation of the harmful bacteria. So that can also increase the risk as well. And then during pregnancy, we find that while the fetus puts pressure on the uterus, it can impact it in such a way so that not all the urine will be voided from the bladder, and so the remaining urine there can also increase the risk of certain bacterial proliferation. And during pregnancy, there can be changes in the hormone levels, which will also affect which type of bacteria can survive, and this could lead to the harmful bacteria proliferating. And finally, we find that during menopause, the vagina and the surrounding tissue can become thin and dry, and that happens due to the loss of estrogen, and that can also make that region more susceptible to urinary tract infection as well. Okay, so those are the things I want to say as far as the various organs of the urinary system are concerned. So now let's move on to talk about the supply of blood to the kidneys. So notice that the renal artery is going to arise from the abdominal aorta. So as the blood is pumped through the aorta, and the aorta travels down to the abdomen, it's known as the abdominal aorta, and then it's going to give rise to the renal artery. And the purpose of the renal artery, as shown here, is going to perfuse the renal pyramids. So these are the structures seen in the renal medulla. And in order to do this, these renal arteries will undergo repeated branching over and over again, as shown here, and this branching will eventually terminate at a tuft of capillaries, and these capillaries are known as the glomerulus. Okay, and this slide does not have a picture of the glomerulus, but this is something I'll show you in the next slide. And we find that the renal veins, they're going to carry the blood which is filtered into the systemic circulation. So from the kidneys, the blood which is going to the rest of the body will be the filtered blood, and it will eventually enter this inferior vena cava and then be taken back to the heart to be pumped to the rest of the body as well. Okay, so this is an overview of how the renal blood supply occurs, and we will talk about this in a little more detail in the next slide. So notice that the renal artery is shown here, and most of these blood vessels have the corresponding vein as well. So here's the renal artery bringing blood to the kidneys, the renal vein taking blood away from the kidneys. Now the renal artery will branch into what are known as segmental arteries, and the reason I've colored this in red is that there are no corresponding segmental veins, right? This is one anatomical difference, whereas pretty much every other artery will have a corresponding vein. And then these segmental arteries, they're going to move in between the renal pyramids, and because they go between the lobes of these pyramids, they're called interlobar arteries. Okay, and you will find alongside the interlobar veins as well. And these interlobar arteries and veins, they're going to arch over the base of the renal pyramids, and so they're called the arcuate arteries and the arcuate veins. And once they arch over the base of these renal pyramids, they're going to radiate into the cortex, and so they're called the cortical radiate arteries and the cortical radiate veins as well. So I hope this naming convention makes sense, why they have a specific name depending on where they are found in the kidneys. Now notice that these cortical radiate arteries, they are going to give rise to an afferent arteriole which will carry blood to the nephron. So this purple structure you see here is the nephron, the basic structural and functional unit of the kidney. And this afferent arteriole is going to carry blood to a tuft of capillaries, and the blood will leave the nephron through the efferent arteriole. So you're already familiar with the term afferent, which means to arrive, efferent, which means to exit or to carry away. So that's the nomenclature here. Afferent arterioles bring blood to the nephron, efferent arterioles will carry blood away from the nephron. So these efferent arterioles, they're going to loop back and they're going to wind around the tubules of the nephron, and so they're called peritubular capillaries, right? So peri means they're surrounding the tubules, so peritubular capillaries. And these peritubular capillaries will lead to a venule which is going to carry the filtered blood to the cortical radiate veins. Okay, so this is how the blood flows, and this representation here will actually explain the direction of the blood flow. So the good news is I don't need you to memorize all of this. What I do want you to know, I have highlighted in yellow, right? And the way you can think about these necessary blood vessels are, what are the blood vessels which bring blood to and from the kidneys? That's the first thing. And we see that the renal artery brings blood to the kidneys and the renal vein takes blood away from the kidneys. This is the first thing. Second thing is, what are the blood vessels which bring and take away blood from the nephron, right, the structural and functional unit of the kidney. So notice here the afferent arteriole is bringing blood to the nephron, and the blood gets filtered and it's going to be carried by the efferent arteriole, and then this is going to circulate through the peritubular capillaries, and then it goes to the venule, and from the venule it's eventually going to be taken to the renal veins as well, right? So these are the only names I'll hold you responsible for, and the reason I want you to know these names is so that you could answer a poll everywhere question like this, right? Which of the following carries unfiltered blood? Is it the venule, the afferent arteriole, efferent arteriole, the renal vein, or none of the above? Which carries filtered blood? This is a fair question. Yes, you can answer it based on that picture given in that slide. Okay, most of you said afferent arteriole, and that is correct, because once the afferent arteriole carries blood to the glomerulus, then the blood will be filtered from that point onwards. Okay, now we are going to talk about the structure of the nephron. This is the structural and functional unit of the kidney, which forms the urine. So if you notice this simplified structure of the nephron, you see that it's kind of like a snake, right? There's the head of the snake, and here's the tail of the snake. And let's talk about the various parts. We see that there will be around 1 million nephrons per kidney, and the nephron has got two main parts, just like how a snake has two parts, head and tail, in very simplistic terms. We see that there is this glomerulus, which is the tuft of capillaries shown here, and that's going to be covered by the glomerular capsule, which is a thin sheath. It's made up of simple squamous epithelial cells. And then there is a small space between the glomerulus and the capsule, known as the capsular space, and you see that here is the afferent and the efferent arteriole shown. So all of these structures jointly are known as the renal corpuscle. And the reason I point all of these structures out is it will all play a role in the formation of urine. So this is the head of the snake, right? Whereas the tail of the snake is the renal tubule, which is going to be this purple part, which is shown as a tube. So this can be divided into the proximal convoluted tubule. So the term convoluted refers to something which is wound round. Tubule means it's a tube-like structure. It's said to be proximal because it is closest to the point of attachment, right? And the point of attachment here is the renal corpuscle. So this will give rise to the loop of Henle, also known as the nephron loop. I'll use this term, these terms interchangeably, and this leads to the distal convoluted tubule. And as the name indicates, this part is distant from the point of attachment, which is the attachment of the tubules to the renal corpuscle. So there is actually one inaccuracy in this representation, right? This is a simplified representation, and we see that in the kidneys, the renal corpuscle will be in close contact with the distal convoluted tubule. It's kind of like the snake is biting its own tail, right? And the reason for this is that if there is something going on in the distal convoluted tubule, that can act as a feedback mechanism to the renal corpuscle, so that according changes can be made, right? So if there is some changes happening in the tail, the head can detect it and it can make the necessary changes. So we find that this distal convoluted tubule is going to lead to the collecting system, and notice that this collecting system is going to receive fluid from multiple distal convoluted tubules, and this gives rise to a collecting duct, which will in turn give rise to a papillary duct, which leads to the minor calyx as well. So you may be wondering, why do we have these different regions labeled in the nephron? And the reason is, we find that if you look at the histological structure, we find that there are different types of cells found in the different parts of the nephron. So we'll see that different types of epithelium will indicate different types of functions in the nephron. So I'm going to quickly walk you through what are these different types of epithelia which are seen. So in the renal corpuscle, notice that that thin sheath which was surrounding the tuft of capillaries is made up of simple squamous epithelial cells, right? So this glomerular capsule is made up of simple squamous epithelial cells. And then when you move to the distal convoluted, the proximal convoluted tubule, it is going to have simple cuboidal cells which have extensive microvilli. And the purpose of this is to have increased surface area so that it can be more effective in what it does. Secondly, notice that there is lots of mitochondria here. So whatever is happening in this region is very energy intensive, and so the mitochondria indicates that it's going to help make lots and lots of ATP aerobically. And then you move down to the nephron loop. Notice that there is a thin descending limb and a thick ascending limb. So this thin part is got simple squamous epithelium, which is got a unique function which I'll talk about, and this thick region has got simple cuboidal epithelium which has another type of function which I'll specify in the upcoming slides. And in the distal convoluted tubule, notice that it's similar to the proximal convoluted tubule. The main difference is that it has got fewer microvilli, meaning that whatever happens in the distal and proximal convoluted tubule are same, but it happens to a lesser extent in the distal convoluted tubule. That's why it's got fewer microvilli. Right? Same things are happening, but it's happening to a lesser extent in the distal convoluted tubule. We come down to the collecting ducts. There are two kinds of cells, but I will not mention it because we will not be discussing that in any amount of detail. And finally, in the papillary duct, we see that it's made up of columnar epithelial cells, and this is the only part where you have columnar epithelial cells. Everywhere else it's either simple squamous or the simple cuboidal epithelial cells. So now let's talk about the various types of nephrons, right? And this is based on the location. We see that the renal corpuscle, right, the head of the snake will always be found only in the cortex, right, and the tail can stick into the medulla, but the renal corpuscle will be found only in the cortex, right? And so we find that there are two types of nephrons based on their location. You have the cortical nephron, which is shown here, and then the juxtamedullary nephron. And so cortical nephrons, they will make up 85% of all nephrons, and we find that almost entirely they are found in the cortex, and so as the name indicates, they are called cortical nephrons. Whereas the juxtamedullary nephrons, they are going to be found at the interface. Notice that the renal corpuscle or the head of the snake is going to be mainly in the cortex, but notice that the tubules are going to be extending deep into the medulla. And notice that these nephrons have got a much longer loops of Henle. They invade deeply into the medulla, and they will play a role in concentrating urine. How exactly this happens, we will discuss in tomorrow's lecture. All right. But here's the question for you: do you think animals which live in the desert, like camels, will they have more of these juxtamedullary nephrons or fewer of these juxtamedullary nephrons? More or less? More, right? And that is true. So camels have around 35% of their nephrons being juxtamedullary nephrons, so that they can draw more water back into the bloodstream and make the urine more concentrated, and that helps them to survive for a longer time without water. Now, a couple of other things I want to mention is that there are these blood vessels which are found in the surrounding the tubules. They are slightly different. So in the cortical nephrons, you have the peritubular capillaries, which I mentioned previously, whereas in the juxtamedullary nephrons, you find that the blood vessels are a lot straighter and longer, and that's known as the vasa recta. So recta means straight, and so you notice that these blood vessels are very straight. So now let's talk about how urine is formed, having considered the various organs of the urinary system and the structure of the nephron as well. So we see that the kidneys will filter the body's entire plasma volume around 60 times per day, and this is a very demanding process metabolically. And we see that the components of the blood will enter the renal tubule, and that's known as the filtrate. Now the urine composition is going to be around less than 1% of the total filtrate, which means that there is a huge effort to rescue the components of the filtrate, and this rescue effort is going to take place in the tubules of the nephron, right? And that's why you find the proximal convoluted tubule has a lot of mitochondria. And the urine will contain mainly metabolic wastes and also unneeded substances as well. So we see that the urine formation is very complex, but it can be divided into 3 simple steps. And let me just orient you to this figure before we talk about the steps in urine formation. So here's the cortical radiate artery carrying the pressurized blood, and as it enters the afferent arteriole, it's going to, due to the blood pressure, we find that the blood plasma is going to be pushed into this capsular space, and then the unfiltered blood will move into the efferent arteriole and move on further. So we see that the first step is glomerular filtration, where we have the passive mechanical process by which molecules which are less than 5 nanometers will enter into the tubules as shown here. Okay, this is the very first step. Now the second step is going to be tubular reabsorption, and it's denoted here by 2 red arrows, and the reason there are 2 red arrows is to indicate that most of this filtrate which enters the tubule is going to be reabsorbed back into the bloodstream as shown here. So what is being reabsorbed back will be 99% of the water, a lot of the amino acids, glucose and other salts in the filtrate as well. Now step 3 is tubular secretion, and if there were some toxins or drugs which were not removed in the filtrate, that's going to be moved from the bloodstream back into the filtrate so that it can be taken out as urine. Okay, so these are the 3 steps in urine formation: filtration, and then tubular reabsorption, followed by tubular secretion as well. So we're going to take this simplified explanation of urine formation, and we are going to see how this fits into the nephron structure. Okay, because this is not the structure of a nephron, it's very simplified. Let's see how it fits into the nephron structure. So we see that in the nephron you have the renal corpuscle shown here, also known as the head of the snake in our class. We find that the filtration will be produced, filtration will take place here, and here's where the filtrate is produced. The filtrate is not produced anywhere else in the nephron, it only happens in the renal corpuscle. And now this filtrate is going to move into the proximal convoluted tubule as shown by this green arrow, and we find that a couple of things can happen in the proximal convoluted tubule. Notice that you have the reabsorption of various substances like water, ions, organic matter, and the reabsorption is indicated by arrows which point away from the tubule into the space, right? So it's showing that substances are reabsorbed into the bloodstream. Now notice that there is this arrow which is pointing towards the tubule, which is indicating secretion, right? And the fact that this is dotted arrows means that this secretion is variable because it depends on what type of hormonal regulation is taking place, and we will talk about a couple of examples of hormonal regulation to indicate whether tubular reabsorption or secretion takes place. As we come down to the nephron loop, notice that the descending thin limb is only permeable to water, right? So there will be further reabsorption of water, and when you consider the thick limb, it's the thick ascending limb, it's going to be permeable only to sodium and chloride ions. So we see that in the thin limb water is being reabsorbed, and then in the thick limb sodium and chloride are being reabsorbed. Now the relevance of this will make sense in tomorrow's lecture, right? Why is this happening? And then moving on to the distal convoluted tubule, we see that reabsorption can take place, but also secretion of ions, drugs and toxins which were not removed in the filtrate initially, they are going to be moved back into the bloodstream through the tubular secretion. And if there is any additional molecules which need to be reabsorbed back, that also takes place in the distal convoluted tubule. Now moving down to the collecting duct, we find that here also there is the variable reabsorption of water and other substances, and we will specifically see how a hormone will regulate this reabsorption of water and thereby make the urine either concentrated or dilute, right? We will talk about this in the upcoming lecture. And finally, papillary duct is going to deliver the urine to the minor calyx, and from there urine will be stored and eliminated as well, right? So this is an overview of urine formation, and when we come back tomorrow, we'll talk about how this filtration is taking place, what are the various parts of this filtration unit, and how tubular reabsorption and secretion take place as well. For now, I just want to go ahead and summarize what we have been discussing, right? We talked about how the urinary system is influenced by various organ systems in the body, including the integumentary system, cardiovascular, digestive, muscular, nervous, and the reproductive system. And multiple organs constitute the urinary system, and I hope you know what are the key differences between males and females and why that increases the risk of UTIs in females. We saw how the blood vessels branch to perfuse the kidneys, and I told you that I need you to know the blood vessels which bring and take blood away from the nephrons. And we saw the structure of the nephron to see how different parts have different functional roles, and we tied that to the formation of urine, which has got three main steps, which is the filtration, reabsorption, and secretion. All right, so I'm going to end class a couple of minutes early, and we'll come back tomorrow and go into more details of the filtration, tubular absorption, and secretion. All right, thank you all for your time.